Welcome to today's lesson on cell differentiation and division. In this session, we will explore how cells become specialized to perform specific functions. Today, we will learn about the process of cell differentiation. Specifically, we will understand how animal cells tend to differentiate at an early stage and explore how plant cells continuously differentiate and divide throughout their life cycle. So far, you have learned that cells can become specialized to perform specific functions. We have seen examples of this with animal cells such as the sperm cell and nerve cell, as well as plant cells like the root hair cell and xylem. This foundational knowledge highlights how various cell types adapt to carry out unique roles within an organism. Now, this understanding sets the stage for us to introduce the concept of differentiation and how it's crucial for both animal and plant cells. This concept advances our biological understanding by showing us how complex life forms develop from a single cell through a series of highly coordinated specialization events. This insight helps us appreciate the incredible diversity and functionality of cells within living organisms. To understand how cells become specialized, we need to delve into the process of cellular differentiation. Cell differentiation is a process where a less specialized cell becomes a more specialized cell type. This involves a series of changes in the cell. These changes can be in cell size, shape, and subcellular structure, which will enable the cell to perform a specific function. For example, as a stem cell differentiates, it might undergo a change in shape to become elongated like a nerve cell, or develop unique structures, such as the flagella in sperm cells. Let's now look at some specific examples of changes in cell size, shape, and subcellular structure. First, let's think about potential changes that could occur in the size of a cell. One example is when muscle cells become larger to generate more force. Now, let's consider possible changes in the shape of a cell. For example, nerve cells develop long extensions called axons to transmit electrical signals over long distances. Finally, a cell may undergo certain subcellular structural adjustments. One possibility is that certain organelles might become more prominent. This is the case for root hair cells in plants. Root hair cells have an increased number of cell membrane folds to absorb more water and nutrients efficiently. Specialization allows cells to perform distinct and efficient tasks that are vital for an organism's survival. Without differentiation, multicellular organisms wouldn't have the specialized tissues and organs that they need to perform complex functions, such as nutrient absorption, for example. In animals, most cells differentiate at an early stage of development. This generally happens during something called the embryonic phase. You can think of an embryo as a blank canvas that through differentiation starts to become a masterpiece. Once animals have grown, their cells lose the ability to differentiate. The consequence of this is that most animal cells are fixed in their roles. For instance, a red blood cell loses its nucleus and will remain as a red blood cell for the whole of its lifetime. After an animal has fully grown, the primary purpose of cell division is to replace or repair damaged cells. For example, skin cells are constantly renewed to replace other dead or damaged skin cells, but they do not switch to becoming any other type of cell. In contrast to animals, many plant cells retain the ability to differentiate throughout the life of the plant. The resulting flexibility is crucial for a plant's growth, as well as its ability to adapt to possible environmental changes. Plants have specific regions known as meristems. These are the sites for continuous cell division and differentiation. Meristems are found at the tips of roots and shoots, thus allowing the plant to grow in length. Before we wrap up, let's explore the history behind the discovery of cell differentiation. The concept of cell differentiation, the process by which cells become specialized for specific functions, began to take shape in the late 19th century. However, the underlying mechanisms weren't fully understood until the 20th century. Key figures in this discovery were the German biologists Hans Spemann and Hilde Mangold.
I conducted experiments on the embryos of frogs and identified what I termed the organizer region, a small group of cells within the embryo that possessed the remarkable ability to influence the specialization of surrounding cells. This work advanced our understanding of how a single cell can give rise to a complex organism and also laid the foundation for developmental biology. This achievement was recognized with the Nobel Prize in 1935.
Together, we conducted the Spemann-Mangold experiment. This experiment was crucial in understanding how cells in a developing embryo organize themselves in order to form a complex organism. The organizer region, we discovered, is now called the Spemann-Mangold organizer.
It's incredible to think that the study of frog eggs has revealed so much about how we develop from a single cell into a complex organism. In today's lesson, we explored the important concepts of cell differentiation and division. To begin, we emphasized how cells become specialized to perform specific functions. We then examined the process of differentiation, which is essential for the development and functionality of complex life forms. This process enables a single cell to develop into a multicellular organism, with each cell type acquiring unique structures and functions. Understanding this is key to appreciating the complexity and diversity of living organisms. We also distinguished between the differentiation patterns in animal and plant cells. We learn that animal cells typically differentiate at an early stage of development. After this, most cells lose the ability to differentiate any further. In contrast, plant cells continuously differentiate and divide throughout the plant's life. This ongoing differentiation allows plants to grow and adapt continuously, forming new tissues and organs as needed. By studying these processes, we gained insights into how specialized cells are generated and maintained, which is fundamental for the growth, repair, and adaptation of organisms. This knowledge serves as a crucial foundation for more advanced topics in biology and helps us to understand how organisms develop, function, and sustain themselves.
Watch the rest of the GCSE biology course at Sophos Education.